the way it's written. It's well known that's what it does. But there are other things, you know, in the cell. And they are competing for some of the intermediates of the Krebs cycle too. So you've got to wait. And oxaloacetate is really, really important. Let's just say now what happens when you have no oxaloacetate. No, we'll call it oxaloacetate, OAA. How would that arise, do you think? Diabetics, undiagnosed diabetics. How about people that are starving? They would have no oxaloacetate. Why would that be? Because in their case, it would be converted to glucose to keep the essential blood glucose level. So there's no oxaloacetate. Now, you know, you must have heard about anorexia, right? Very bad disease. So, I've had several students die from this. And actually one of our postdocs died just a few days ago. She looked terrible. She came to a seminar in October and she looked like she was dead already. So anorexia the origin of anorexia is not known. It's obviously something to do with brain chemistry, but it's not been diagnosed. They don't know how it works. And to treat it is really, really difficult. So in my freshman seminar, I take pictures of all the kids, you know, the 19, maybe I can remember who they are, that's the main point. So I took a picture of this young lady, and 4 days later she's dead. So I had the last picture of this child, and I sent it to the parents. So why would you die from anorexia? And then there was another one in biology 1107. And then actually the daughters of one of our faculty members also had anorexia. And very hard to treat. And it's very dangerous because people don't think it's dangerous, but what do you suppose it will kill you? Take a guess. What is it that you don't want to do as far as your heart is concerned? What do you need as an ion to control heart pumping? Potassium. So, you know, an anorexic maybe eats food, but then takes laxatives or vomits. And what happens then is you screw up the electrolyte balance. You know, the amounts of sodium, potassium, phosphate, all that. And pretty soon you get too low in the potassium and your heart stops. When it doesn't stop, it gets irregular, so it doesn't pump. That's what happened to that young lady, and actually the one in biochemistry. So, it's something you take very seriously. It's not that, you know, some kind of joke. Anorexia is a dangerous, dangerous disease. It's really difficult to treat. Very difficult to treat. Okay, but now let's say, as far as this situation, somebody is starving or diabetic. So we're getting all these acetyl-CoAs. We're getting them from sugar, from proteins and fats. They're building up. They have nowhere to go. They can't enter the Krebs cycle, which is the ideal solution, because when they enter the Krebs cycle, we're going to get all those nice ATPs and all those nice NADHs. But now they have nowhere to go. So as the acetyl-CoAs build up, you get what are called ketone bodies. One of these ketone bodies is acetone. And I think I mentioned to you that if somebody has a sweet pungent odor, if somebody is an undiagnosed diabetic, I mean, you know when somebody's starving, right? So that's pretty obvious. But diabetic, they will have acetone in their breath, in their urine. Does acetone belong in your body? No, bad stuff. It's an organic solvent. It will dissolve membranes, stuff like that. Another thing that's formed is acetoacetate. And the third thing is beta-hydroxybutyrate. Now we're going to go into a lot of detail about this, but what you should know is that when there's no oxaloacetate, ketone bodies form from acetyl-CoA. There are acetone, acetoacetate, beta-hydroxybutyrate. Now, acetone you know is bad, but actually, in moderate amounts, these two compounds are metabolized. Your heart likes these two compounds in moderate amounts. They'll metabolize them. But we're not talking about moderate amounts, we're talking about huge amounts. So both these things have a carboxylic group. Both these compounds have a carboxylic group. So as the concentration builds up, and you can't metabolize it, it builds up, gets into your bloodstream. Now what happens when you have a lot of carboxylic acids? pH will drop. pH starts dropping, and you go into acidosis. Does that sound good to you? Acidosis is not good. You go into a coma, and you could die. So, it's a bad disease. Diabetes is bad, but once as soon as you tell these people that there's something wrong about you, you need to go to the emergency room, they can treat that. Diabetes can treat starvation, that's a different story. Okay, so as a result, we got to have more ways of making acetylacetate. It's really crucial. So here's an enzyme. I think we may have talked about this before. It's called pyruvate carboxylase. And acetyl-CoA is a positive regulator. This is an allosteric enzyme. Acetyl-CoA is a positive regulator. Why would that be? Because as the acetyl-CoA builds up and has nowhere to go, it's going to signal this enzyme to go to work. Okay? And what this is going to do is, it's going to take pyruvate, it's going to pick up pyruvate, instead of going to acetyl-CoA, it's going to take up the pyruvate, react it with CO2 and ATP, and it's going to make acetylacetate. So this is the direct way of making acetylacetate, in making sure the Krebs cycle is working. Important step. Okay, here's another enzyme. This is called a malic enzyme. Don't confuse it with malic dehydrogenase. This is a different enzyme. And this is using NADPH as a reducing agent. So again, pyruvate plus CO2 plus NADPH is going to reduce this carbonyl oxygen to CHOH. You can make L-malate, and L-malate can enter the Krebs cycle and make acetylacetate. So this is a little bit indirect. Finally, the third way, which you've also seen before, and where do you think you've seen this step before too? It's the reversal of glycolysis. Gluconeogenesis. But here is pointed out again that this is really important, because this is another way of making acetylacetate. So it takes PEP, which was formed in glycolysis, takes CO2 and GDP, and you make acetylacetate. So there are at least 3 different ways, eventually, of making more acetylacetate to make sure the Krebs cycle works, and that does not. Those series of reactions, we'll be talking about them in detail. We don't want to get those ketone bodies. Finally, in the Krebs cycle we should now look at where all the ATPs are made. Okay? So, we're going to make 38. Let's start with glycolysis. So glycolysis goes to 2 pyruvates, and we make 2 NADH, right? Thinking back to glycolysis. So the 2 net ATPs are 2, and then each NADH is worth 3 ATPs after it undergoes oxidative phosphorylation. Now the trouble here is glycolysis is taking place in the muscle. We're not in the mitochondria. So to get that NADH into the mitochondrial area, you're going to have to need a shuttle system that talks about the various shuttle systems. We're just assuming that nature is very efficient. We're going to use the best shuttle system, and we're not going to lose any ATPs. Then, what we just talked about Wednesday, this pyruvate oxidase complex, it makes acetyl-CoA plus CO2 plus NADH. There are 2 NADHs. 2 NADHs. Each NADH is worth 3, that's 6. So when you add these up, 2, 6, and 6, you get 14 ATPs. Now the Krebs cycle goes to work. The Krebs cycle goes to work. And what happens in the Krebs cycle is the following, right? All the steps we just talked about. So at the isocitrate, the alpha-ketoglutarate, you made an NADH. That's worth 3. When we were talking about the alpha-ketoglutarate oxidase dehydrogenase complex, we made an NADH. That's worth 3 ATPs. Then we went from succinate-CoA to succinate. You know, that was a high energy bond. We hydrolyzed that and we made a GTP, which is worth an ATP. Then we went from succinate to fumarate. We involved the ubiquinone, and we made UBH2. That's worth 2 ATPs. Finally, the last step, mainly going to oxaloacetate, you make another NADH that's worth 3. You add them all up, and you get 12. 12 ATPs. So 2 acetyl-CoAs, that's what goes through, actually, from glucose. Each acetyl-CoA is worth 12. 2 times 12 is 24. You add these all up, and there I go 38. How about that? Each adds up. So, all the reactions that we talked about, they're all fair game. You're not going to be asked to regurgitate the Krebs cycle or glycolysis. But a reaction we picked out, and you need to know the structures, the reaction to product, and you have to know what it does. Understand what it's actually doing. So don't blindly memorize all this stuff. You're going to get screwed up in the exam. Any questions? Okay, we'll start reviewing the following.